So the lumen of the gut, the GIT, contains trillions of microbiota, which exceed the host cell by 10-fold, and the genes that are expressed by this microbiota also exceed the host gene expression by almost 100-fold. Normally, complexity of the gut microbiota is generally associated with defense against pathogens. And commensal microbiota have diverse functions, and to mention a few, they have a metabolic, structural, and protective functions that they provide to the host, and the host provides them with a very conducive environment for their growth and also proliferation. Very little is known about the interplay between viral pathogens and commensal microbiota, but recent studies suggest that viral pathogens can significantly alter the composition of gut microbiota. And as you can see here in mice, some of the most important microbiota microbes were suppressed with infection of high pathogenic influenza virus, the top, and then in chickens with Marek's disease virus, a shift in the composition of the microbiota was observed. And in macaques infected with SIV, the same individuals showed a disrupted gut microbiota after infection. And this is not limited only to composition of microbiota, but a recent study also showed that vaccine efficacy was also importantly impacted by the lack of commensal microbiota. And co-injection with flagellin, which is a major component of the commensal microbiota, was able to rescue the TIV-specific antibody response within 7 days of vaccination. And this was dependent on TLR5. So we hypothesized that commensal microbiota play a critical role in the initiation of antiviral immune response against AIV infection in chickens, and also anti-AIV vaccine efficacy in chickens. And we did a preliminary study whereby we infected chickens at day 16 with H9N2, and we collected the cecal contents at day 1, 3, 5, 7, and 9 post-infection, and we did a quantitative real-time PCR to characterize some selected microbial populations to sort of have a bird's eye view of what's happening. And we saw that early on in the infection, some of the microbiota are upregulated, and also some of them are downregulated. And we further did a principal component analysis to see if this disruption is actually occurring between infected and non-infected chickens. And we saw that the non-infected groups were clustering together, but while the infected chickens, they showed a more dispersed microbiota composition. And then we did another study, which is in progress in terms of the next generation sequencing, to be able to see an in-depth analysis of what's actually happening in the gut microbiota, because QRT-PCR is not going to answer a lot of our questions. And the other part that we are doing is to try to be able to see the association between microbial composition and viral infection. In this case, AIV infection, you have to be able to try to deplete the microbiota. And one of the important techniques that have been used for a long time is germ-free animals. But for our case, because of its high running cost and maintenance cost, it hasn't been feasible. So we set out to develop a new model in chickens by trying to use a cocktail of antibiotics to deplete the microbiota. And this has been recently used in mice. And we tested this to try to develop the model. And we were able to deplete the microbiota, both the culturable in terms of aerobic and anaerobic microbes. You can see that there was no growth of microbes in the antibiotic-treated chickens. And we saw about 50-fold decrease in the composition of the total bacteria in antibiotic-treated and non-treated chickens compared to non-treated chickens. And this was not at the cost of the body weight or any overall physical appearance or health of the chicken. We saw actually an improved body weight in the antibiotic-treated chickens. So we have a model set up in our lab now. The next step is to try to evaluate the gut and respiratory system associated innate and adaptive immune response of chickens with induced dysbiosis and challenged with H9N2. And with this, we'll be able to determine the microbiota-associated immunomodulatory mechanisms in the control of AIV in chickens. And also we'll study the role of gut microbiota in AIV vaccine efficacy in chickens. And then we'll be able to make recommendations of candidate microbes for future use as vaccine adjuvants or probiotics against AIV infection in chickens. With this, I would like to acknowledge funding agencies and my advisor and advisory committee and members of the Sharif Lab. Thank you.